all my wonderful KCET champions. I hope you are doing absolutely great, children. Well, today we are finally going to end the chapter Principles of Inheritance and Variation by dealing with the last part of it, that is NEET plus KCET PYQs, that is previous year question. So are you ready for a double treat where we will be seeing how different type of questions come from both of these examinations? If at all you're ready, as of now, just now, click on the like button. And my dear children, make sure you have a pen and a paper with you and you try to solve the questions by yourselves as well. So we are over here, we started from here and we came until this point, and I am super proud of all of you who have been with me throughout this journey. Okay, and I am guaranteeing you, if you were with me, then 30 marks for sure in your PU2, 4 to 5 questions for sure in your KCET examination. Now, going ahead with this particular very important thing, that is, so we have made up this slide where we are talking about the rankers who scored really well the previous year. We are talking about the hard work that they do and things like that. We generally talk about them in every single video. Now, I want a favor from you, and that favor is, I want you also to put enough hard work such that I can talk about you in the next year as well. And you know what, whenever we talk about these children out there, we are immensely proud of everything that they have done, and we are really happy that these guys are someone that you all know, and I want you guys to be in this position the next year. So work really hard for that. Now going ahead, starting off with our game. Okay, I'll talk about the instructions. So first of all, read the question carefully and attempt the questions by yourselves first. You should not be depending just on me. You have to try it from your end. And in case you get your answers wrong, please note down the question number and revisit the question number later on. You will get the PDF as well. Okay, you do not need to worry. No cheating. Please go through all the lecture videos. If you go to the lecture videos, I can guarantee you, you will be able to get all the answers. If there is a new concept question that has been added up over here, I'll take the responsibility to explain you here itself. Okay? Chalo. So starting with the NEET questions, I have few NEET questions out there. The first one, among the following characters, which one was not considered by Mendel in his experiment on P? So Mendel chose 7 characteristics. I hope you remember that. So it is said that which one among them is not under those particular 7 characters, or does not come in the 7 characteristics which he studied. So trichomes, glandular or non-glandular, we have not studied about this thing ever. So the first one itself is something that is out of the box. Seed, green or yellow, that has been studied. Right, green and yellow, that has been studied. Pod, inflated or constricted, children, you should not remember this, but it is also one particular character. Stem, tall or dwarf, that is the most famous character. So please remember all the 7 characters which are given in your book. Okay, flower color, flower position, pod color, pod shape. Okay, pod color and pod shape, and then seed color, seed shape, and the last one is what? The height, that is over here. Perfect. The 7, please remember them. All which is dominant, which is recessive, that also you have to remember. Children usually get confused between green and yellow. So in pod color, green is the one that is, yellow, wait. Even I got confused. Yeah, green is the one that is dominant. So we always pick, when we go to buy peas, right, we always look for the green one. Have anyone picked up yellow ones? No, right? So green one is the dominant one. Now when we open it, we see the pea. The seed color is usually green. The ones that we take is usually green, but the dominant one in seed color, it is yellow. So in pod color, dominant one is green, but in seed color, even though we have green seeds only, but it is actually yellow one which is the dominant one. Please remember that also. Okay, now going ahead. The second question, identify the wrong statement with reference to the gene I that controls ABO blood group. So there's a gene I that controls ABO blood group. You have to identify which statement is wrong. So I'll read out the question. You attempt from yourself. If you think that I am going in a high speed, pause the video, try it by yourselves. Okay. So the gene I has 3 alleles. Does it have 3 alleles? Think. Okay. A person will have only 2 of the 3 alleles. When IA and IB are present together, they express same type of sugar. Allele I does not produce any sugar. Well, my dear children, gene I has 3 alleles. Yes, IA, IB and IO, or just I. And the person will have only 2 of the 3 alleles? Yes, will have only 2 of the 3 alleles that you see. And now when IA and IB are there together, they express the same type of sugar, is it? No, they express different type of sugar, but both of them are able to express themselves. So yes, they express different type of sugar, not the same type of sugar, which is why this becomes the incorrect statement. Allele I does not produce any sugar. The small I, what we call as IO, it does not produce any sugar. The answer, that is also correct. This is correct. It does not produce any sugar. Only A and B produces sugar, and A is dominant over O, B is also dominant over O. A, B are co-dominant over each other. They produce different type of sugar, not the same. So this one is the incorrect one. Cool? Now, in Antirrhinum of snapdragon, a red flower was crossed with a white flower. So red and white flowers were crossed, and in F1 generation, all the flowers were pink. Actually, we thought that red is something that is dominant, so all the flowers should be red. But over here, we see it is pink in color. So pink comes in between red and white, right? When pink flowers were then selved and produced F2 generation, it showed white, red and also pink. When pink flowers, these pink progenies which were crossed, my dear children, then you got white again, you got red also, and you got pink also. Choose the incorrect statement from the following. Law of segregation does not apply in this experiment. This experiment does not follow the principle of dominance. Pink color in F1 is due to incomplete dominance, and this ratio is obtained in F2 generation. What is the answer? Well, first of all, here, this experiment does not follow the principle of dominance. It is correct. Actually, it does not. If capital R was completely dominant on small r, it would have been red, but now it is not red, it is pink in color. So yes, it does not follow law of dominance, so this statement is correct. Pink color in F1 is due to incomplete dominance. This process is known as incomplete dominance, so this is also correct. Ratio of F2 is that 1 out of 4 is red, 1 out of 4 is white, and 2 of them. So this is capital R, this is small r, this is capital R, small r. So these would be pink. 2 out of 4 would be what? Pink in color. Law of segregation does not apply in this experiment. That is wrong. Law of segregation means what? For example, this is the parent plant. Capital R, small r, right? Both of them will separate. Only one will be going in each gamete. Similarly, here also only one will be going in each gamete. When this and this comes together, you get capital R, small r. So aren't they segregating? Yes, my dear children, they segregate. So law of segregation does apply over here. It's not like it does not apply. That is a wrong statement. So this is the one which is incorrect. Okay, here since it is does not apply. Now, the genotypes of a husband and a wife are IAIB and IAIO. The small I means IO. Among the blood types of their children, how many different genotypes and phenotypes are possible? Genotypes means genetic makeup. Phenotypes means physical appearance, my dear children. Okay, so let's try to solve it. So we have, is it visible? Or we'll write it here. So we have IAIB and then we have IAI. Fine. So if you cross this and this, you get IAIA. What do you mean by IAIA? That means A blood group is there. Okay. Now we cross this with this, another genotype IA and small I, or basically IO. That means again A is dominant over O, so A blood group. This and this, we get IA and IB. That means AB blood group, since both of them are co-dominant. Now the last one, IB and small I. B is dominant over small I, so it will be B blood group. So how many different phenotypes can you observe? AA. Here both of them are A, so I'll consider it as one only. A, AB and B. So 3 phenotypes are possible. 3 physical appearances are possible. Now what about the genotype, my dear children? This is different, this is different, this is different, this is also different. So 4 genotypes, 3 phenotypes. 4 genotypes, 3 phenotypes would be your answer for this question. Got it? Now, test cross involves what? Crossing between 2 genotypes with dominant trait, crossing between 2 genotypes with recessive trait, crossing between 2 F1 hybrids, crossing the F1 hybrid with a double recessive genotype. What is the answer for this? So test cross means I am unaware what is the genotype of an organism. Okay, so whether it is capital T or capital T small t, I do not know, because anyway both of them will be tall only. Both of them are going to be tall. Here since both of the alleles are same, here since it is dominant, which is why it will be tall. Now for knowing this, for understanding if the F1 hybrid that I have, the F1 progeny that I have got, my dear children, what is the genotype of it? What will I do? I'll try to fuse it or mate it with the double recessive genotype. I'll try to fuse it or mate it with what? The F1, what do you call the F1 parent out there? Not the F1 parent. Generally, we'll do a back cross. We'll cross it with a parent with recessive alleles. With recessive alleles. So that will be what? Small t, small t. When I cross it, I'll get different type of progenies here. And if I cross it with this, I'll get different type of progenies here. When I cross this and this, all of them will be tall only, because here I have 2 capital T. So whatever combination do, at least 1 capital T will be coming with small t. Okay, all of them will be like this. So all of them will be tall. But when I cross and this, my dear children, some of them will be dwarf and some of them will be tall. So this is what I mean by test cross. And in that way, I'll get to understand it is tall like this or it is tall like this. So crossing the F1 hybrid with a double recessive genotype is known as test cross. If at all, in the question it was given, crossing the F1 hybrid with the double recessive parent also, it would have been fine. Okay? Cool. Why don't you cross with dominant parent? Because the dominant parent will show its own trait. So in that case, we won't be able to identify what is the trait of this. Okay. Now, 6th question. A new question. Okay. It was not taught in the class. So listen to me carefully. What map unit or centimorgan is adopted in the construction of genetic map? What map unit is adopted in the construction of genetic map? A unit of distance between genes on chromosome representing 50% crossover, a unit of distance between 2 expressed genes representing 10% crossover, a unit of distance between 2 expressed genes representing 100% crossover, a unit of distance between 2 genes on the chromosome representing 1% crossover. So not to make it too confusing, I'll tell you the answer directly. When you say 1 map unit or 1 centimorgan, okay, when you say centimorgan, 1 map unit, by that we mean that there is 1% frequency of what? Recombination. 1% recombination frequency is there. If I say 2 centimorgan is there, that means what? 2% recombination frequency is there. That is what I mean. It has nothing to do with the distance and stuff like that. It might confuse you. So just understand, it is showing actually when you're talking about what map unit is adopted in the construction of genetic maps, it is basically the unit of distance between genes on the chromosome representing 1% crossover. So this would be your answer. Okay. Now, in a test cross involving F1 dihybrid flies, more parental type offsprings were produced than the recombinant type offsprings. This indicates what? So you understand one thing, no new recombinations are found, only the parental one, the older, just like the parents, the progenies are being born. By that, what do we understand? That means, my dear children, not too much amount of recombination is happening. Not too much amount of new combinations are being found. Why? Probably because the existing genes which are there, they are very close to each other. They are very close to each other. So even if the chromosomes are swapping here and there their parts, the genes anyway, they are very close to each other. So they are also moving together only. So there is no formation of new character. So what do we understand from this is that the genes which are there, they are tightly linked. They are tightly linked. When they are tightly linked, there will be so less distance between them. Always understand this very, very important point. We say there is more amount of recombinant type offsprings when the genes are very far away from each other. So suppose this part of the chromosome went there, this part of the chromosome went here. So one gene is over here, now we are getting a new gene on this side because there's a swap happening. But since they are very far away from each other, which is why you get new combination. But here that's not the case. They are so close to each other, they are tightly linked. That means they have lesser distance between them. Now, so your answer would be what? In a test cross involving F1 dihybrid flies, more parental type offsprings were produced than recombinant type offsprings. This indicates that the 2 genes are linked and present on the same chromosome. Both of the characteristics are controlled by more than 1 gene. The 2 genes are located on 2 different chromosomes. The chromosome failed to segregate during meiosis. Your answer would be the 2 genes are linked and are present on what? The same chromosome, which is why they are not showing any new combinations up there. So A. Cool. Now, There are 3 genes A, B and C. Percentage of crossing over between A and B is 20%, B and C is 28% and A and C is 8%. What is the sequence of genes on chromosome? Now I have seen that biology children, they run away whenever they see numerical terms out there. But trust me on this question is so simple. It is super simple and you have to be there with me. Okay, I'll explain you how you can answer this properly. Okay. So I hope you're ready. Let's begin. So they have mentioned about 3 genes. What are those 3 genes? A, B and C. And they have given us the crossing over percentage of each of them. And they are asking what is the sequence of the genes on the chromosome. In what sequence these genes are arranged on the chromosome. That is the question. So my dear children, between A and B it is 20%. Okay, between B and C it is 28%. So first tip is that whichever thing has too much distance, you keep them in the extreme. So B and C, I am keeping them in an extreme because they have what? 28% crossing over. That means they are showing high amount of crossing over. That means they are far away from each other. Now A and B has 20%. A and B. So A, let's put it towards C because A and B should have distance, right? So I'll put 20%. Now, A and C is 8%. A and C, if you see, they just have 8%. Now, you tell me, 20 plus 8, 28, right? So, what will be the sequence? B, A and C. It will be B, A and C. So, your answer would be A, my dear children. Okay? It's easy. Trust me. It's B, A and C and this is how you work. First of all, you look for the highest term out there and then you do the rest of the things. Cool. Okay. 10th, in human beings, multiple genes are involved in the inheritance of what? Multiple genes. Usually we study that basically one gene is responsible for one trait. But there is something that is known as polygyny, where multiple genes are there which will be deciding the trait. And where do you see it? Sickle cell anemia, skin color, color blindness, phenylketonuria. Polygyny, where do you see it? You will be seeing it in skin color. Which is why we all have different skin tones, my dear children, because multiple genes are expressing themselves. And depending on how they are expressing, we have so many shades of colors. Okay, so your answer would be skin color. Even human height is an example of this. Okay, human height is also an example of this particular thing. Cool, let's go ahead. Match the terms in column 1 with the description in column 2 and choose the correct option. Dominance, codominance, pleiotropy, polygyny. Dominance means what? Basically, whenever there are 2 alleles, for example, 2 different alleles. What do I call this condition? Heterozygous condition. Whenever there is a heterozygous condition, if one is able to express over the other, this is dominance. So let's see that. In a heterozygous organism, only one allele express itself. This would be what? Dominance. So A goes with 2. A goes with 2. I have only 2 options. Let me delete all the other options. Let me eliminate those options. Now, codominance. Whenever you have 2 alleles, for example, A and B, in the case of blood group, both of them are able to express itself. But are both of them same? No. Again, in a heterozygous pair, both of them, when they are able to express themselves, it is known as fully, okay, completely. It is known as what? Codominance. So, in a heterozygous organism, both alleles express themselves fully, it will be codominance. If they are expressing partially, incomplete. Okay, so B goes with 3. B goes with 3. I have this option. So anyway, this would be my answer now. Anyway, let me check. Pleiotropy. What do you mean by pleiotropy? There in polygyny we studied multiple genes are there for one trait. Now suppose one gene is responsible for multiple traits. Then this is known as pleiotropy. Okay, so many genes govern a single character. That is what? Polygyny. Many genes, polygyny. Okay, and pleiotropy goes with a single gene inheritance influences many characters. That is pleiotropy. So D goes with 1. D goes with 1 and C goes with 4. C goes with 4. So your answer would be this. Cool. Now, number of Barr bodies in XXXX female S. Now this is again a new thing. How do we answer it? Let me tell you. So basically when you say XXXX, understand very important thing. When a woman is born, she has XX chromosome, right? Her both X chromosomes are not active. One X chromosome becomes inactive and they turn into Barr body, which looks like a spot in the nucleus, my dear children. So in this condition, XXXXX female, only one has to be active. Okay, rest 3 of them would become what? The bar bodies. And that is what you are being asked. Number of bar bodies in XXXX female is what? It would be 3, because you need 1. Rest all of them will become a bar body. Cool. Now, in the case of males, if you are asked, in the case of males, how many bar body do you have? None. Why? Because this X chromosome is there, that will be active. Okay, there is nothing to get inactivated because there is no other X chromosome, there is another Y chromosome. So in males, there is no bar body, and that is how sex determination is being done. In the nucleus, if you see bar body, that means it's a female. If you do not see the bar body, that means it is a male. So over here in this condition, it is 3. Number of bar body is what? 3. Okay, I think that is a new information for you guys. Okay, now, which of the following most appropriately describes hemophilia? Hemophilia is what? A blood disorder where what happens? A gene coding for a clotting enzyme, my dear children, a clotting protein that which is involved in the clotting process is not being produced. It is mutated. The gene is mutated. In that particular condition, even if you get a small cut, you will immensely bleed forever. So that is a problem. Okay, that is the thing. So over here, which of the following most appropriately describes hemophilia? What would be your answer for that? You do not have blood disorder, anything, ma'am. You have genetic things given over here, and I do not know. So it is actually a X-linked recessive gene disorder. It is what? X-linked. That means it is on the X chromosome. Okay, it is on the X chromosome, and it is recessive gene disorder. For example, if a woman has a condition like this where her one X chromosome gene is mutated, okay, but the other is fine, then in this condition she would not have hemophilia, she would be just a carrier. Only in recessive condition, that means when both of them are same, okay, and that condition only she will be hemophilic in nature. So that is what you mean by X-linked recessive gene disorder. Here in the case of hemophilia, males are the ones that are mostly affected. We studied in our lecture videos. I hope you know why. Because males have only one X chromosome. There is no other X chromosome. There is only Y chromosome. There is only one Y chromosome. So if the X is affected, it will show that trait. Because there is no one to mask the effect of X. Okay? Because it is like a recessive condition. Now, yeah. Okay. Now this question, I want you to pay lot of attention. Okay, so in the following human pedigree, the filled symbols represent the affected individuals. Identify the given pedigree over here. So first of all, you have to understand whether it's dominant or a recessive thing. X-linked autosomal, we'll come to that. X-linked means it is on the sex chromosome. Autosomal means it is the gene which is affected, it is on the other chromosome apart from the sex chromosomes. You call them autosomes. What do you call them? Autosomes. What do you call the sex chromosomes? Allosomes. So allo and auto is different. Okay. So over here, you see that in every generation, someone is getting affected. In every generation, this is generation 1, generation 2, generation 3, generation 4. Whenever you see this, when every generation things are being repeated, you understand one very important thing, that this is a dominant disorder. Because in dominant disorder, there is no skipping of generation. Generations are not skipped. In recessive condition, that's when the skipping of generation is seen. In dominant, it is not seen. Now, it is autosomal. It is what do you call autosomal, or is it sex linked? How do we identify that? So my dear children, again, if in first look, if we see, if it is sex linked, usually when you talk about sex linked, mostly the males are being affected. But over here you see the father is being affected, and then here the son is also being affected. Right? The father is being affected and the son is being affected. How can that happen? In the case of what do you call X-linked dominant. Anyway, it's dominant, we got to know that. So first of all, if we try to decode this, suppose if it is X-linked, so the X chromosome would be having this particular problem, and the female over here, since she is not affected, she can be in a condition like this. But if she is in a condition like this, my dear children, when the X and the Y comes together, the male should be normal. Now suppose she is a carrier. Suppose she is a carrier. Okay. In that condition, my dear children, over here you need to understand that here, since we are talking about dominant disorder, in this condition, the female also should have been diseased, right? Because only one chromosome is needed, one affected chromosome is needed for the person to be having the disorder, because this is a dominant disorder, my dear children. So in this case, the female should have the disease, but that is not the case over here. You get it? So basically it means that it has nothing to do with the X chromosomes or its X chromosomes. It would be what? Autosomal dominant disorder. It will be an autosomal dominant disorder. That's all. Okay, here mostly the males are affected, but that does not mean that's true. And you got to know why it's not true. Because here she cannot be a carrier. At least you have to be a carrier. If it is X-linked, the mother had to be a carrier for the child, the boy, to be having the disease. But over here, there is no term for carriers because it is a dominant disorder that we are talking about. So this would be what? Autosomal dominant disorder. Example of autosomal dominant disorder is myotonic dystrophy given in your NCERT. Okay, that is what? Myotonic dystrophy. Now, yes. A man whose father was colorblind marries a woman who had a colorblind mother and a normal father. What percentage of the male children of this couple will be colorblind? So first of all, we know one thing, that the father is colorblind. Okay, so father is colorblind means the condition is like this. Now the mother, we do not know what's her condition. So for getting to know the mother's genotype, we have to cross her parents. So woman who had a colorblind mother means both the X chromosomes should be like this, because it's a recessive, X-linked recessive disorder. Colorblindness is a recessive disorder. So both the chromosomes should be like this. And a normal father. For the father to be normal, he should be like this. Now, when I cross this and this, I get she to be the carrier. When I cross this with this, again I get she to be the carrier. If I cross this with this, I get the boys. I do not need the boys as of now. I'm talking about the mother. So mother, either way, would be definitely a carrier. Okay, whatever daughters are born for them, all of them are carriers. So over here, I'll take the mother to be the carrier. Now we have to find what percentage of the male children of this couple will be colorblind. So basically, again, we have to cross. Let's cross all of them. Okay, this and this, you get the female carrier. Why she is a carrier? Because she just have one chromosome which is affected. This and this, she is a female with color blindness because both of them are affected. This and this, that's a male, a normal male. This and this, that's a male who has color blindness. So out of two males, one male will definitely be colorblind. So can I say 50% of the male will be colorblind? I'm not talking about all the kids. Here, what percentage of the male children of this couple will be colorblind is asked. So in out of two, one will be definitely colorblind. So that means 50%. Out of two, one means 50%. So your answer would be this. Cool? Cool. Chalo. Now, Down syndrome in humans is due to what, my dear children? In Down syndrome, what happens? The 21st chromosome, okay, you have 3 copies of it. That means one extra. Usually we get one from the father, one from the mother, but here you're getting one extra copy of it, which is why there are 3 chromosomes. That's how you get the disease, that is Down syndrome. What do you call this? Trisomy. You call this condition as trisomy. And here your answer would be 3 X chromosomes. No, it is not X chromosomes. It is the 21st chromosome. That means you're talking about autosomes. Sex chromosomes are the 23rd chromosomes. Okay, please remember, 23rd pair of chromosomes. So over here, autosomes would be, okay. Down syndrome in humans is due to 3 copies of chromosome number 21. Now, which one of the following symbols and its representation used in human pedigree analysis is absolutely correct? Which one of the following symbols and its representation used in human pedigree analysis is correct, my dear children? So, box and then 2 lines and then circle mating between relatives, is that correct? Unaffected male, unaffected female, male affected, which one is correct? So, here circle means female, so this is wrong. Here it is male. Here square means male, here it is female, so that is wrong. When this type of thing comes up, that means the sex is not specified. Okay, and it is the sign of affected, but since it is colored, but the sex is not specified. Here it is written male, so this is also wrong. So we are left with this, which means this is the correct one. Mating between relatives. When 2 lines are there, that means a very strong relation. You can think like that. It is known as consanguineous mating. Okay, Nice. With that, we are done with all the NEET questions out there, and now we are going ahead with the second set of exam, that is what? KCET. And I have taken 10 questions for KCET. So let's do that. The first one. In one of the hybridization experiments, a homozygous dominant parent and a homozygous recessive parent. Homozygous dominant parent means capital T, okay? Dominant. Homozygous also same alleles. And a homozygous recessive parent are crossed for a trait. So when we cross them, what happens? You tell me first. When you cross them in F1 progeny, all of them will be tall, right? Now in F2 progeny, what happens, my dear children? There will be both tallness as well as dwarfness. So over here, when I cross them, all of them are capital T, small t. Whatever I do, I will get capital T, small t only. Whatever cross I do. Now if I cross between them, what will I get? Capital T, small t, capital T, small t. If I cross between this and this, then I get capital T, capital T. If I cross between this and this, capital T, small t. This and this, again capital T, small t. This and this, small t, small t. So basically in the second generation, I got the dwarf ones and the tall ones. In the first generation, all of them were tall. Tall is what? The dominant character. Now you tell me. Dominant parent trait appears in F2 generation and recessive parent trait appears only in F1 generation. Wrong. Recessive will be appearing in F2 generation. Dominant will be appearing in both F1 and F2 generation. Dominant parent traits appear in F1 generation and recessive parent trait appears in F2 generation. Wrong. And children usually take it correct. Don't do that. Okay. Dominant trait appears both in F1 and F2 and recessive parent trait appears only in F2 generation. So your answer would be C, my dear children. Okay. Now, which of the following statement is correct? Female carrier for hemophilia may transmit the disease to sons. Female carrier for hemophilia may transmit the disease to sons. Do you think so? So female carriers means the condition is like this. Now the father will have Y. Over here you get the Y chromosome. Yes, in this condition, female carriers can transmit the disease to the sons. This is true. The first statement itself is correct. Thalassemia is a qualitative problem. Is it? No, it is a quantitative problem. Depending on how many alleles of beta globin gene or alpha globin gene is getting affected, that is how it is a quantitative problem, not a qualitative problem. Change in the whole set of chromosome is called aneuploidy. Is that so? No. Change in the whole set of chromosome is known as what you call polyploidy, not aneuploidy. Now, sickle cell anemia is a quantitative problem. No, it is a qualitative problem. Beta globin gene which is produced, the quality of it is bad. Okay, so that is the thing, and which eventually makes the hemoglobin also bad, which eventually makes the RBC structure also really bad. So this is also, all of the other things are wrong, only the A1 is correct. Okay. Find the correct statement. Generally, a gene regulates a trait, but sometimes one gene has effect on multiple traits. One gene has effects on multiple traits. Yes, pleiotropy. Okay, where do you see it? In phenylketonuria. There is a gene for phenylalanine hydroxylase. This helps in the conversion of phenylalanine to tyrosine. So when this enzyme is not produced, this conversion does not take place. Okay, and when this conversion does not take place, my dear children, we actually needed tyrosine for the production of melanin. So melanin is also not produced. If melanin is not there, your skin pigmentation, your hair pigmentation, everything becomes reduced pigmentation. Okay, all of it will be having reduced pigmentation. So that is what you mean by hypopigmentation. So first the character that it takes is that tyrosine is not being produced, which is why, so that means conversion is not happening, which is why phenylalanine stays like that. It converts into phenylpyruvic acid and causes mental retardation. That is the first thing. Second thing is that since conversion is not happening, since tyrosine is not being formed, melanin is also not being formed, and the skin pigmentation and everything gets disrupted. Okay, so one gene regulating multiple traits, that is true. That is known as pleiotropy. Now, trait AB blood group of a man is regulated by one dominant allele and another recessive allele, hence it is codominant. Wrong. Okay, first of all, you have three alleles. Okay, there is dominant allele, that is A, and B, both of them are dominant over what? O. But when A and B come together, both of them are codominant. That means both of them, there is no one dominant and one recessive. Both of them would be equally dominant. That is what you mean by codominance. And over here, there is a whole different statement given. So that would be wrong. The first statement is the only one that is correct. So statement 1 is correct. Now, going ahead, example for non-Mendelian disorder. thalassemia, hemophilia, cystic fibrosis, Down syndrome. Example for non-Mendelian disorder. What do you mean by non-Mendelian? Non-Mendelian disorder. Any guesses? Any guesses? Basically, the ones which are not, what do you call, caused because of genetic mutation or mutation of the genes, okay, those are known as non-Mendelian disorders. Thalassemia, it is a Mendelian disorder. It's because of the gene being mutated. Hemophilia is a Mendelian disorder, gene being mutated. Cystic fibrosis, if you talk about that, it is also what do you call, because of the mutation of a gene, okay. What about Down syndrome? Down syndrome is caused because one extra chromosome is being added. It's not because the gene is getting mutated somewhere. Okay, so this is an example of non-Mendelian disorder. That means it is a chromosomal disorder. It's a non-Mendelian disorder. Okay, so your answer would be D. Now, the fifth option. Match the column 1 with column 2. You have autosomal trisomy, allosomal trisomy, allosomal monosomy, cystic fibrosis. So, autosome, I told you, out of the 23 pair of chromosome, 22 of them are autosomes. Only one pair is the sex chromosome, which you call as allosomes. Okay. Now, when you say autosomal trisomy, that means in the autosomes, one of the autosomal pair, you have one more chromosome being added up. It's a trisomy condition. Where do you see it? In Down syndrome. I just told you 21st chromosome, that's a trisomy. So, A goes with 4. A goes with 4, I have 2 options. Let me eliminate the other 2 options. Now, allosomal trisomy, that means sex chromosomes, instead of having 2, usually we have 2 only, XX, if boy is there, XY. But instead of that, one extra X chromosome is also coming, or once some chromosome extra is coming. This is known as trisomy condition. And when it is happening with the sex chromosome, you call this allosomal trisomy. So, where do you see it among them? It is Klinefelter syndrome, where instead of having XY, you have XXY. There is an extra X chromosome. This is known as allosomal trisomy. So, B goes with 3. B goes with 3. Again, you have option like that. Now, allosomal monosomy, where do you see that? Allosomal monosomy is basically where sex chromosome is there. So, instead of having 2 of them, X and Y or X and X, you just have 1. That is allosomal monosomy. One chromosome is being deleted. So, this happens where? In Turner syndrome, where you have XO condition. Instead of having XX, okay, one chromosome is not there. You didn't get one chromosome from the gamete out there, okay, from one parent. So, it is just XO condition. So, C goes with 1. C goes with 1. Now, I have cystic fibrosis. That is a Mendelian disorder. That means it's a genetic disorder. It's not chromosomal. So, this would be your answer. Okay. Chalo. In an organism, mutation in a gene, in an organism, mutation in a single gene exhibits multiple phenotypic expression. Identify the underlying genetic mechanism in the above instance. Mutation in a single gene. When a single gene is mutated, multiple phenotypes are getting affected. What is it? Pleiotropy. I told you, a single gene mutation, a gene is controlling multiple traits. This is an example of pleiotropy. Multiple allelism means multiple alleles are there. For example, IA, IB, IO. That was alleles. Incomplete dominance means one allele is not completely dominant over the other. Polygenic inheritance means multiple genes are required for one particular trait. That is polygenic inheritance. So, your answer would be pleiotropy. Okay? Okay. From the chromosomal combinations given below, identify the one which shows female heterogamety. From the chromosomal combinations given below, identify the one which shows female heterogamety. What will be your answer for this? Female heterogamety means females are the one which produces 2 different type of gametes. For example, if we talk about human beings, here the females which are there, they produce only one type of gametes. All of them will be having X chromosomes. If we talk about males, either X or either Y. So, over here you have male heterogamety being seen. 2 type of gametes are being produced by the males. If you are asked about female heterogamety, there are birds which show that. Okay, there are birds which show that, and in them it is ZZ, ZW. So, ZZ, that means same type of chromosomes. This is for males. ZW, this is for females, my dear children. Females produce 2 type of chromosomes. One this and the other one is this. Okay, so your answer is this. You should know the other ones, XX and XO type. Where do you see it? In the case of insects. Okay, in the case of insects, you see that this is also an example of male heterogamety, either X or no chromosomes at all. Okay. Now, in the following symbols used in human pedigree analysis, identify the symbol that denotes consanguineous mating out there. Consanguineous matings mean, I told you, mating between relatives. So, now children know that double line means mating between relatives, but they do not know this term. Then you will lose your marks. So, please understand, it's given in your NCERT, you should know it. So, what is your answer? Your answer would be B. Okay. 9th, sickle cell anemia is due to the following mutant gene. Sickle cell anemia is where point mutation is an example of point mutation also. That means only one single base pair is getting changed. So my dear children, instead of having GAG, these are nitrogenous bases. When they come together, they code for an amino acid, my dear children. And when such amino acids are being grouped together, it forms a protein. So over here, instead of A, it is replaced by GUG. Instead of adenine, nitrogenous base, another nitrogen base, that is uracil, is being coming up there. There's a point mutation at a point, which is why instead of forming this amino acid, which is known as glutamic acid, we are forming some another amino acid which is known as valine. And now amino acid is changed, right? So the protein produced will also be somehow different. You get it? It will be somewhat different. So this is the what do you call, when you talk about sickle cell anemia, this is the case. Now my dear children, where do you see GAG, which is what, where do you see GAG? You see it in the case of mRNA. You see it in the case of mRNA. This mRNA is being read to make the proteins. Where do you get the mRNA? How is mRNA being formed? When you read the DNA, that's how you get the mRNA. We'll study in the next chapter. So in the DNA, the sequence would have been C, T, C in normal condition. Okay, but the DNA, in the DNA, the mutation took place and now it was CAC. Which is why whenever RNA was being formed, it will make the code like GUG. I know it might be confusing, but listen to me carefully. A always pairs with T, G always pairs with C, and the same goes here also. Okay, that is in the case of DNA. So you have double stranded DNA. So in one strand, suppose it's A, in the other strand it will be T on the opposite side of it. Now when you make an RNA out of the DNA, my dear children, okay, A is not going to pair with T. In RNA, you have something else which is known as U, and U will be pairing with A. Right. So when you make the when you make RNA out of the DNA here, so C goes with G, T goes with what? A, and C goes with again G. So it should have been GAG. And over here, since the mutation has taken place, here C goes with G, but A will be going with what? U, my dear children, and C will be going with G. So actually the mutation takes place in the DNA itself, and here you have an option like this. But in general, okay, in general, you majorly talk about the mRNA, not the DNA. So children get confused, between this and this, but please consider this to be the answer. Okay, because generally we consider mRNA, and that is given in your NCERT. That is even more important, right? In your NCERT, it's exactly given about this. They have not mentioned about the sequence of DNA. They have mentioned about the sequence of mRNA, which is like this. Okay, and always remember A comes before U, so it was A first and then it got replaced with U. Similarly, G comes before V, so it was glutamic acid first, then it got replaced to valine. Okay, so your answer is this. Now, the 10th one, choose the possible genotype responsible for the lightest skin color in human beings. Now, this is the last question, which makes it more interesting, right? So, for the light, I told you, for skin color, you have multiple genes. Gene here, gene here, gene here. Suppose it is capital A, capital A, capital B, capital B, capital C, capital C. In this case, what is the genotype of this organism? AA, BB, and CC. This has the darkest skin color. If a person has this genotype, the darkest skin color. Now suppose it's not capital A but small a. Okay, in that case, capital A, small a, capital B, capital C, capital C. This will be lighter than this. That means again, if it is small b, then it is capital A, small a, capital B, small b, capital C, capital C. This will be lighter than this. Suppose this is small c, then capital A, small a, capital B, small b, small c, sorry, capital C, small c. This will be lighter than this. What will be the lightest one? When all of them are small. That's when you get the lightest one. So over here in the option, which one is the one? It will be C. Small a, small b, small c, small c. Okay? Okay. Fine. With that, we complete all the questions for this particular chapter. Obviously, there are more questions. We will be doing it more towards the examination. We are not seeing much of the audience being watching the video somehow. Probably you are very, you're waiting for the examinations to start to study. If that's the idea, please do not have that. But anyway, if you are someone who is watching until this particular point, I understand you have taken it seriously and you have started it from this particular point of time. But if you have some friends who have not started yet, ask them to study as well. That's your responsibility, okay? And wishing you good luck, my dear children, and lots of love. Take care of yourselves. Bye-bye, take care, and have a happy Sunday tomorrow. Bye-bye.